Welcome to MOOC NPTEL course on Bioengineering, an interface with biology and medicine. So, today we are going to talk about cell cycle and in which way cell cycle dysregulation may cause diseases like cancer. We are going to talk about why mitosis is so important, why meiosis is central to reproduction. We are also going to look into various factors, internal and external factors which regulate cell cycle. We are going to talk about the difference into the normal and cancer cells and what is the basic molecular biology research in which we have started investigating about the cell and its properties to try to find some clues for cancer. Let us first start with cell cycle. So, as you can see, the life begins from a single cell starting from the sperm and egg cell, their fusion in the process of fertilization that give rise to the embryo, and from embryo from the one cell now after the continuous division the life starts. The cell division distributes the DNA of a mother cell equally between two daughter cells, therefore, it allows a cell to proliferate. This cell proliferation is very crucial for the growth, repair and the reproduction process. So, what is the cellular organization of the genetic material? So, if you think about broadly a genome actually that consists of the entire DNA molecules in case of eukaryotic cells, and if you think about prokaryotic cell context, it is a single DNA molecule. Now, let us also get some definition for somatic cell. These are the normal body cells, non-reproductive cells which contains two set of chromosomes, whereas the gametes they are the reproductive cells which are especially sperms and eggs, and they contain half as many chromosomes as the somatic cells. So, let us now talk about cell cycle. In the cell cycle, the mitotic phase alternates with a large growth phase which is the interphase. Now, this entire cell cycle could be divided into multiple phases. Let me go to board and draw you the cell cycle. Alright, so the cell cycle, one of the phases of the cell cycle is known as G1 phase, and then we have S phase and G2 phase, and then we have a short mitosis phase. The mitosis phase is much smaller, but rest of the part which is essentially the interphase or the growth phase, that is much bigger phase, right. So, the entire mitosis process which is going to be responsible for cell division to happen, that is actually much shorter phase, but remaining phase which you can see, that is known as interphase, that is much larger phase. So, this is interphase which prepares the cell and ensures that now cell is ready for division to happen. So, the mitotic phase alternates with much longer interphase, and it is almost 90% of the overall cell cycle which comes as a part of the interphase. Now, the G phase names actually comes little misnamed, especially the gaps, but these are not the gaps, these are actually quite crucial for cell to prepare, and G1 phase ensures that all the nucleic acid contains the chromatins that sufficient present, and then now cell is ready for DNA replication to happen in the S phase, and then the G2 phase is there which now ensures that the DNA replication has occurred properly and cell is ready for further division to happen in the mitotic phase. So, now the same concept you can see in the slide that we have G1, S and G2 phase. The G1 phase is first part of the interphase, S phase is where the chromosomes get duplicated, the G2 phase is last part of the interphase, and M phase is where the mitotic division happens and this process distributes the chromosomes to the daughter nuclei. In the process of cytokinesis, then the cytoplasm get divided from the in the two daughter cells, and now the two cells contain the same cytoplasm, and now they have a bifurcation from the parent cell. So, let us look at broadly the mitosis and meiosis processes. In case of mitosis, the division of somatic cells happen whereas the meiosis governs the division of gametes, especially the sperm and the ova. In mitosis, the two daughter cells which are produced, they contain the same amount of DNA as the mother cell. In case of meiosis, 4 daughter cells are produced which contains half the amount of DNA as the mother cell. Let us first start discussing about mitosis and why mitosis is so important. So, think about all our the growth requirement. Many times if the old cells get replaced, old cells get replenished, the damaged cell has to be repaired and new cells has to be produced. All this only happens because of the process of mitosis, which is the process in which the cell divides to form 2 identical daughter cells. Let us first talk about few basic terminologies before we actually talk about the mitosis process. So, for example, in the blue color on the left side, you can see a pair of sister chromatids are there, and then we have this DNA which is hidden behind this the protein complexes. That region is known as centromere, and the protein which is surrounding to that is kinetochore, which is the protein attached to the centromere region. And this chromosome has the pair of sister chromatids. So, the 2 chromatids are shown in the blue colors. Now, let us think about a cell which is having 3 chromosomes each from father and mother. Let us say the maternal set or the mother obtained chromosomes are shown in the red color, and the blue one is shown from the father. So, the 2n or the ploidy level is 6 chromosomes in this case. So, each chromosome is having these sister chromatids, which whether it is in the blue ones or the red ones, which are connected with the centromere. And now, when they form the homologous pair, that is both the blue ones and the red chromosomes comes together, that is the chromatids from the non-sister chromatids of the homologous pairs are being formed. Or you can say that these are pair of homologous chromosomes from both one from each of the paternal and maternal sets is shown in the circle here. Another interesting fact is that how DNA is so so much DNA content is present, but how it is so tightly packed in the nucleus of every cell in such a short space. So, it is not only DNA, but in addition to DNA there are separate histone proteins which are also attached to the DNA molecules, and these 8 histone proteins along with the DNA molecules, they form a complex which is nucleosomes. And these the fiber of these packed nucleosomes that is known as chromatins, and these chromatins they get condensed before the process of mitosis starts. So, in the image shown here is various histone protein like histone H1, we have histone H2A, H2B, H3 and H4, and you can see that how 8 histone proteins are packed up and forming the nucleosome, and many of these nucleosomes together will form the overall chromatins. So, let us have the broad overview of mitosis process first. So, thinking about again the overall cell cycle, we have this mitosis phase and we have the interphase. Now, within mitotic phase, then there are different phases involved. For example, prophase, prometaphase, metaphase, anaphase, telophase and cytokinesis. Let us go each one of them in slightly more detail one by one. So, first let us look at thinking about this nuclear content, they are getting condensed and then they are forming the chromatins, right. So, that is what is shown in the blue color here. The sister chromatids are being formed and chromatin, the condensed material is forming now the discrete chromosomes, and during this process the nucleoli get disappeared from the cell. So, each chromosome appears as 2 sister chromatids and they are actually joined at the centromere. Now, then at the same time the centrosomes they move apart and the mitotic spindle starts to form. Now, you can see this particular electron microscopy image which kind of shows that how people make observations in the actual research looking at the cell images, and that is what is then drawn in the cartoon form. Now, let us move on to the next phase, which is prometaphase. Now, cell is trying to prepare itself for division to happen and start orienting it towards the 2 poles, and that is the time when now nuclear membrane starts fragmenting and the microtubules they starts growing. Then each of the two chromatids which have the kinetochore proteins at the centromere, now these microtubules get attached to those kinetochores and they are known as the kinetochore microtubules. So, again a lot of shuffling is happening in the cell. All these the chromosomes and these chromatids from these chromosomes start trying to align themselves towards the metaphase plate, which is just in the center, but they have still not aligned. They are trying to align themselves, but then the cell moves to the next phase, which is metaphase. Now, at this time the chromosomes they get assembled at the metaphase plate, which is the equidistance between the two spindle poles. If you think about from two spindle poles from the top, then just in the center that equidistant region is where the metaphase plate is formed. So, each chromosome sister chromatids they are actually attached to the microtubules which are arising from opposite poles. Now, you can see the same in the microscopic image. Alright, so now let us move on to anaphase. During the anaphase, the sister chromatids they separate, and now each chromatid they behaves as a chromosome. So, during this process things are not so straightforward. A set of protein, the complex proteins are involved, especially the cohesins they help in the process of cleavage, and then the daughter chromosomes they move towards the opposite poles. Now, a similar concept now can be seen in the microscopic image on the screen. So, then the last part comes, which is the telophase, and then the cytokinesis happens. So, for example, two daughter nuclei form in the cell. Now, the nuclear envelope start reappearing because they want to now produce two daughter cells. The spindle microtubules they get depolymerized, and then the chromosomes becomes less condensed at this time. And at the same time now the karyokinesis process or division of nucleus also gets completed. So, the microscopic image shows you that now from one cell now the two cells started appearing, although the distinct boundaries are still not made, and that happens in the process of cytokinesis. So, at the process of cytokinesis these cell furrows being formed, and now the division of cytoplasm gives rise to two daughter cells. So, now let us kind of review the whole thing again. We talked about prophase, we talked about metaphase, then metaphase, anaphase, telophase and cytokinesis. So, this complete the how from one cell the two daughter cells are being produced having the same nuclear contents, and that kind of ensure that body is growing, there is a sufficient development is happening. Now, let us move on to another important process, which is meiosis. So, what is meiosis and why meiosis is important? So, think about if the cells are dividing, and in the case of meiosis, if the numbers of the chromosomes are getting reduced half, and if the process is so mechanical, then every offspring from the same parents will look exactly same, right. So, if let us imagine that there are 4 children from in one family. So, from the same father mother, then they will look exactly same, but there is actually a lot of genetic variation. So, question is from where this genetic variability comes. So, the ability to generate these kind of genetic variations in the children, because the process of meiosis happens randomly, and at that time lot of gene shuffling happens across the chromosomes, and that actually ensures that each one of us is very unique in our appearance, in our identity, in our entire genetic components. So, meiosis is very central to the reproduction process, and if you look at the cell here, we have starting from the sperm and the ova, which is produced inside the testis, the sperm, and inside the ovary these eggs are being produced. Now, these are haploids shown in the green color. After fertilization process, they form the diploid zygote, which is now the 2n. So, what we can see the ploidy level here, that one phase is having haploid level, and now the other part, when after the fertilization and the mitosis and development which happens, that is the diploid level. Now, the multicellular diploid adults are being formed, which are having 2n equals to 46 chromosomes. So, let us now move on to meiosis, but considering the time constraint and thinking about what is so crucial for you to understand, let us not talk the entire meiosis process in detail, but rather try to compare and see what are the salient features which are different in meiosis as compared to mitosis. So, mitosis we have talked in some detail and you have some good idea now. We will now compare that what are the key differences of mitosis and meiosis. So, for example, in meiosis as opposed to mitosis, there are two divisions happens, meiotic 1 and meiotic 2. Now, meiosis 1 having certain peculiarity, but meiosis 2 is exactly like the mitosis. So, now let us take the same parent cell which we took in the beginning for the definitions purpose, which has the 3 red and 3 of the blue chromosome from maternal and paternal sets. Now, let us review again the mitosis process. For example, the chromosome duplication happened. Now, you can see these the condensed these chromatids on the left side in the prophase, 3 blue ones and 3 red ones. Now, these duplicated chromosomes, they are forming 2 sister chromatids. Then after the pro-metaphase and the metaphase, now these individual chromosomes get lined up at the metaphase plate, which is equidistant from the spindle poles. And now in the process of anaphase and telophase, the division happens and now 2 daughter cells are produced, which are having 2n exactly the same like sex chromosomes where you started, right. That is what the left side of the image conveys, which refreshes your understanding for mitosis. Now, let us try to compare a few things in the meiosis. So, in meiosis, now as you can see, the blue and red chromosomes, they are having some sort of interaction, and in certain region there is a crossing over happens. So, this crossing over is known as the chiasma, which is the site of crossing over, and that is the time when some of the genes start getting shuffled from the blue to the red ones. So, from the maternal and paternal side, now we have few genes which are going to shuffle to both the chromosomes. And now these homologous chromosomes, they are actually pairing and they are still held together by the chiasma, and now the sister chromatids by the cohesin proteins. So, this entire thing is slightly different as compared to what you see in the prophase of the mitosis. So, let me highlight the 3 distinguishing feature here which makes meiosis more unique. The very first one was the crossing over or the formation of chiasma. Now, these homologous pair of the chromosomes, they are itself moving and they are forming the metaphase plate when they are actually getting lined up at the metaphase plate. So, in this case now not the individual chromosome, but rather the pair of the homologous chromosome, they are reaching to the metaphase plate, metaphase 1. And then now these homologs, they get separated when the anaphase 1 and the telophase 1 processes are happening, and therefore, now the daughter cells which are produced, they are having the both red and blue patterns together, right. And then further once you have finished your meiosis 1, then the entire process is of mitosis getting replicated, and on meiosis 2 you are going to produce 4 cells out of 2 cells. So, then you have daughter cells of meiosis 2 which are 4 cells now, and n becomes half. So, you have only 3 chromosomes, but each of the 3 chromosomes, as you can see, you do not have very clear red or very clear blue, you have the patches of red and blue, which means the genes have shuffled and the genetic variability has been introduced. So, to just summarize the first part of the lecture, we have first tried to understand the cell cycle broadly, the very small part of mitotic phase which is so crucial, and how the cell divide to form the 2 identical daughter cells. We have also tried to understand that how meiosis generates the genetic variability in the offsprings. And now, I think we are going to continue our understanding about the cell cycle, but more in the context of its regulation, dysregulation, and how the disease like cancer may happen. So, now let us talk about the cell cycle regulation, its deregulation, and how disease like cancer may happen. First, let us look at the cell cycle checkpoints. Just imagine that when the cell cycle is being operated, you want certain checkpoints to ensure that the entire cell cycle is governed properly. Just imagine that when you are driving on a road, you need those kind of the speed breakers, you need the traffic police to ensure that everybody is obeying those rules. Otherwise, sometimes things may go unattended and accidents may happen, right. So, like that there are certain checkpoints even in the cell, and there is no police inside the cell. So, there are certain proteins which are following the same pattern of what you to regulate these particular signals. So, the cell cycles are actually having the checkpoints which are constituted by the proteins which regulate cell cycle by giving either stop or go ahead signals. Then there are different checkpoints involved. For example, even in the G1 phase there is a checkpoint or a section point which ensures that now the the chromatin material is sufficient, the cells are getting condensed with those nuclear contents, and it is getting ready to move to the S phase for DNA replication to happen. Then there is a G2 checkpoint which now ensures that now cell is totally ready for the mitotic division to happen, and then mitotic checkpoint is there, or the M checkpoint is there, which ensures that the mitotic division has happened properly. So, as you can see, there are different checkpoints which are involved to ensure this process is happening sufficiently. Now, how these things happen? So, as I already mentioned, the G1 phase, the parental DNA strands are intact before DNA replication begins. So, the G1 phase checkpoint ensures that this process is done properly. Then the G2 checkpoint, it ensures that newly synthesized DNA strands, they are complete and intact before the mitotic process to happen. And then in the metaphase plate, the M phase checkpoint, the chromosomes are aligned properly at the metaphase plate before the anaphase happens. So, this is kind of various checkpoints which are so essential for the correct distribution of complete chromosome set between two daughter cells. Now, let us think about this entire intricate process which is so complicated, how it is being governed by certain proteins, right. So, a set of proteins known as cyclins and cyclin dependent kinases, they play an important role to govern these processes. Let us now go back and in our cell cycle diagram, now let us draw that how these proteins where they are being synthesized. All right. So, the cyclin proteins are synthesized in the S phase, right, and now they are getting accumulated as they are moving into the G2 phase, and then at that time they are interacting with CDKs or cyclin dependent kinases to form a complex which is CDK and cyclin complex. Now, this ensures now that cell is now ready to move for the entry in the mitotic process. Now, once the mitosis is finished, then the cyclins get degraded, and now this complex get dissociated. Now, the CDK that separate and cyclins got degraded. So, this ensures that now the mitotic process has finished. So, in this way, just by using these proteins, cell is ensuring that this particular cell cycle is governed properly. So, let us look at this in this diagram now. The synthesis of cyclins happens in the S phase. Now, the cyclins are getting accumulated, and then the cyclins are forming a complex with CDKs. Then they are allowing the cell to enter in the mitotic process, and then the degradation of cyclins happen and CDKs get dissociated, which ensures now this cell has completed that particular part. So, this now moves the cell into the G1 phase. So, these are the family of cyclins and CDKs which are very crucial for cell cycle regulation, and these CDKs or cycle dependent kinases, they are expressed throughout the cell cycle. However, they are activated only after when they are bound to the cyclins, which could be synthesized during the S phase and gets accumulated in the G2 phase. As I mentioned, that cyclins get degraded after metaphase, and now the cell enters in the G1 phase. So, in addition to these, there are various external signals which also regulate cell cycle. So, what could be some of the internal signals and external signals? I think let's discuss about them. So, for example, nutrients, growth factors, even space and the substrates, all of these things are also ensuring in which way cell cycle is regulated. So, just imagine that nutrients and growth factors are of course crucial for the cell cycle to operate, but even otherwise even the space can be constrained, like how much space is available. If it is too crowded and there is not much space, then cell should ideally stop dividing, and that is a process known as density dependent inhibition, because there is not enough space now for more cells to grow, and that is where the difference comes to a controlled growth versus uncontrolled growth. In case of normal cells, now normal cell will stop growing when they see this kind of density has been achieved, then density dependent inhibition is happening. Whereas, in case of cancer cell, they are not going to get affected, they are not going to obey this rule, and they keep continuing to grow, and they are then going to form the tumor mass. Now, additionally, these cells are going to attach to certain environments, certain substrates for their anchoring, and that is known as anchorage dependence way for growing the cells. So, again this part is not being obeyed by the cells in case of the cancer cell. So, now let us look at what happens in case of the cancer cell, which they lose dependence on the internal and the external signals for the proliferation. So, cancer cells, they do not obey the normal rules for the cell cycle. They will not stop at the cell cycle checkpoints. They will not exhibit any kind of density dependent inhibition, and therefore, they will form the lump, the tumor mass with a multiple layer of the cells. They do not require any anchorage with a specific substrates to get attached, and therefore, they can actually grow much happily on any environment, and now they can also they are uncontrolled in growth. They are not obeying the cell cycle checkpoint. So, therefore, they are actually in some way they are not at all following the normal cell cycle rules. So, there are a couple of food for thoughts from a journal which shows here that how do healthy cells become cancerous, and these are still an area for research. We have to still think about why the cell growth is problem in cancer, how long do cancer cells live for, why are the cancer cells so powerful. So, I do not have time to talk about each one of these points. I will give you reference. I think you should read this paper, and you should try to understand that how much challenge we have still in science, and to do research in this area we have still not understood these cell properties so much. Why do not cancer cells die normally? Why we cannot control them? What are the missing checkpoints in these cancers, and how do cancer cells they escape destruction? So, all of these are the food for thought for you, and I think you have some reference to read, but there is still too much research to be done in this area. Now, what is important again to emphasize that there is so much need for you to know the basic biology, but also implement that at the technology level, that we need detection technologies, accurate detection technologies to detect the cancer cells early. So, there is a lot of emphasis on looking at even circulating tumor cells with the new detection technologies, and in the United States Food and Drug Administration, the US FDA, they have cleared some technologies for which allows very sensitive capture of circulating tumor cells, which is looking at certain antibodies against the EPCAM or epithelial cell adhesion molecules, which are coated with certain ferrofluids. I am showing you again from a review article two different approaches here. One is immunocytological technologies, and second is the molecular based technologies, in which way by looking at certain markers, some of the proteins which are found in these tumor cells and an antibody generated against them, how this understanding, this knowledge could be utilized to build the devices which could detect the circulating tumor cells ahead of time and much more accurately. So, these kind of technologies will be required. I am again going to give you a reference to read this paper and not talking in detail right now, but just illustrate that basic understanding, basic science is revealing the right targets, the right proteins against which the antibodies could be generated, and then the technologists have to come into the play to find out a way to develop the devices, the detection system which could accurately detect these cells. And is there a way now, just imagine the challenge for you, can we now if out of let us say 1000 cells, one of the cell is showing you the tumor behavior, can you with any kind of magnetic property, can you exclude that out from the normal cells? So, then can we now control the cancer if we are having these kind of technologies which can detect the tumor cells, which can also exclude them out of the normal cell circulation. These are the challenges for us, but I think in an area which needs lot of exciting research. So, in general today in conclusions we have discussed about broadly about the cell cycle and various mitotic and meiotic phases, the kind of contrasting features of mitosis and meiosis, different internal and external factors which govern the cell cycle. I just trying to kind of convey you some basic differences between normal and the cancer cells. And I am sure you appreciate that there is a lot of fundamental research going on to look into various type of the gene, mRNA and protein level changes in normal and cancer cells to find out the right marker proteins which could lead us to find out the good detection technology to detect the tumor cells. And I have shown you a couple of food for thought, some challenging questions in the area of cancer biology, and also some of the promising technologies which are coming forward which could be very helpful in the translational research in future. Thank you very much.